want to choose 2, like 5 and 6, or 6 and 7, or 5 and 7, and then you answer all 3 afterwards. So let's go ahead and say we're going to do 5 and 6. Why not? And start outline the functions of enzymes. So 4-point question, I believe. So enzymes speed up reactions, that is, they catalyze them. Each enzyme is specific for that certain type of substrate, that lock and key model. Enzymes lower the activation energy and are not changed during the reaction, so they can be reused afterwards, recycled. And to work, the substrate needs to bind with the active site that is specific for it. So just outlining the basic functions, what they do. If you do a little graph, this is the activation energy needed. Enzymes lower that, so you don't need as much energy going in. The next question for number 5, explain the roles of specific enzymes in prokaryote DNA replication, which is very similar to eukaryotic replication. Start out with, and it mentions specifically, I believe, enzymes, so you want to focus on the enzymes. So you can start with DNA gyrase. That helps to uncoil the DNA, kind of loosen it up a little bit. Then the DNA helicase comes in and unwinds the DNA by breaking those hydrogen bonds at the base in between the 2 strands. And then the next step, we have DNA primase come in there. And they add a DNA or RNA primer. It's so interesting that to make DNA, you need RNA. So kind of makes you think probably RNA was here first, right? Kind of the evolution of molecules. Then a DNA primase adds an RNA primer. Then DNA polymerase 3, that's the workhorse, adds nucleotides to the template, creates a new complementary strand. DNA polymerase 1 then goes and removes the RNA primer. You don't want that RNA still there. You want to get rid of it. And the DNA ligase joins the Okazaki fragments together that are on that lagging strand. Remember, the leading strand goes along smoothly, right along with the DNA. The lagging strand, little fragments. Remember, the direction is always 5 to 3. The direction of anything, replication, transcription, translation. If you're moving along a deep piece of DNA or RNA in any cell, it's going to be going in that 5 to 3 direction. And then you might want to add the DNA then proofreads for mistakes later on. DNA polymerase 1 and 3 proofreads the DNA for mistakes. The next part of that question, talking about genetic diseases due to recessive alleles. The code for an enzyme, still focusing on that enzyme. Using a Punnett grid, explain how parents who don't show signs of disease can produce a child with the disease. So check out my Punnett square here. You can see, say both parents, they're carriers, so they don't show signs of disease. They wouldn't necessarily know they had the disease, in their alleles or in their genes, unless, they knew about a family history. And you'll see when these genes are separated through independent assortment during meiosis, and then go into individual sperms and eggs, and they can recombine. You can see homozygous dominant, heterozygous, and then homozygous recessive. And this is the offspring that will have the disease. There won't be any dominant gene like the parents have to block the action. And there's a 25% chance that the child will get that disease. Alright, I think I just finished this one. So, Punnett square. Review those. So now I'm on to question number 6. Label diagram. Ooh, I like diagrams showing the positions of the proteins. So we're focused on proteins. This is what you want to definitely get into. You have the phospholipid bilayer. I have a carrier protein here with like molecules coming in and out of the cell. This could also have been a pump protein, right? The sodium potassium pump, looks similar to that. Just make sure you label it. I think they mentioned label diagrams. So yeah, make sure you label your diagrams. I normally force you guys to do everything labeled. Anyway, here's the integral protein going through. Here is a peripheral protein. It kind of sticks on the end, and this could be like an immobilized enzyme. Or it could be a signaling protein. Like a nice little cascade, biochemical cascade. Glycoproteins are on the outside. Maybe you can label this outer, this inner. And if you put a glycoprotein, you want to say, label the carbohydrates attached to it. So that make you good, make you whole for that part A. Now we got part B. What would happen if you put plant tissue in a hypertonic solution? So you've done this, I believe. The red onion cell lab. Hypertonic solution has more solutes than the plant tissue. Water will out of the cell into the hypertonic solution. Water wants to move towards a higher solute concentration. And for some reason, water moves towards a lower water concentration. But they don't want to hear that. I went and looked at the key, and they don't want to hear that. I really don't understand why. But that pressure inside the cell will drop as the water, like the air going out of a balloon, and that plant will become kind of floppy, kind of like when we did the, you guys did or we all did the little potato slices in the quarters, put them in the fresh water and then salty water, they got pretty floppy. And then something might be good, for example, if you put a red onion cell in hypertonic solution, you're real salty. Remember those cells got darker because the cytoplasm shriveled up, it lost volume, and that concentrated the pigment, so it looked darker, a darker color. You can also say the cell membrane receded from the cell wall, so we saw that gap between the two. Now the next question, looking at proteins, 6. Structure of a nephron and how the associated blood vessels enable it to carry out its function. So structure and function is what we want to look at here. Keep that in mind as we're writing our responses. I believe it is 7 or 8 points. 8. So it's a big one. So you want to write some stuff. Something's 8 points. So first, I want to put this. The function of the nephron is to remove urea, nitrogenous waste from the blood. Kidneys made out of hundreds of millions of these little tiny nephrons. Ultrafiltration happens at the glomerulus. That's the little fist ball of cells at the beginning of a nephron, and it's surrounded by the Bowman's capsule. So you have high pressure. The structure, you have a large vessel coming in, small vessel going out. That creates high pressure, and that forces small things out, small molecules out of the glomerulus. So we'll say small vessel leaving. I did reverse. Large vessel entering. So you got all this blood coming in, but not a lot of blood coming out, and that has the effect of raising inside the glomerulus. And what's also good, the structure, you have a thin basement membrane and special sieve-like cells called podocytes that help keep large molecules in, like proteins, and allow smaller molecules to leave, like salt, water, glucose, urea, toxins. Then you have a coiling of the tubule of the nephron that gives it more surface area, more, just space to reabsorb substances. And this is where it's like salts, glucose, and water. Water is reabsorbed in the descending loop of Henle, as well as in the collecting duct. The longer the loop of Henle, remember this is structure, the more water can be reabsorbed. So the nephron structure is different based on the function of what it needs to handle. So desert animals is going to be long, and the rainforest is going to be short. So desert animals, long loop, and rainforest is going to be short loop. And the cells of the collecting duct respond to the hormone ADH. So they, structurally, they will respond, and they will get more porous if ADH is present. So more water can be reabsorbed. So that's if you did number 6 or number 5 or 6. Let's now do number 7. All right, so number 7. So say you did 5 and 7 or 6 and 7. Let's do 7. Draw a labeled diagram of a eukaryotic plant cell. Here it is. There's some features you want to point out. The chloroplast, you'll see double membrane, so 2 lines. You want to let them know, that chloroplast has a double membrane. Grana, these thylakoids stacked up, so these granum stacks. Most plant cells have a pretty significant vacuole. That's one of the characteristics of a plant cell. And then, of course, you have the cell wall here. You want to label that, and then the inside membrane. So I drew a 3rd line. I probably shouldn't have done that. So if we draw it again, this would be the cell wall. It's pretty thick. And then you could draw this part of the membrane. Just that very, very inside. They want to know, I think, that that cell wall is thicker. Then you can draw the nucleus. That will be visible in your electron micrograph. And it'd be good to also show that's a double membrane. Any eukaryotic cell, nucleus is double membrane, and it has pores. That's what these little marks are. Label it nucleus. Mitochondrion, you got the double membrane too. And cristae, the little, I could have drawn a little bigger, I guess. It's kind of run out of space though. Also double membrane. Hey, why not throw in the Golgi apparatus? Okay. So that would be a nice plant cell to place in there. And the next question. How the energy flow through food chains limit their length? So you start out, that 90% of the energy is lost as you go from one trophic level to the next. That is from the plant to the primary consumer, 90%. Primary consumer to the secondary consumer, 90%. So you lose energy really fast. And released by respiration, lost is heat. And also some parts of the plant or the animal, nobody can eat it. Nothing can really get at that food. And even if you do eat it, no animal is 100% efficient in converting it into energy, usable energy. So by the time you get 3 or 4 steps along that food chain, most of the energy is gone. And I just put this diagram here, kind of shows a good example, I think. start out with 1,000 kilocalories, then you go to 100, then 10, and then 1. It just happens really quickly. So you really can't, you just lose so much energy so quickly in a food chain. And the last one, hot, dry conditions, plants losing water. How does the plant replace this water? So this would be like your exam. I think I gave this exam question to you on the topic 9 exam. So the water evaporates from the leaf. Pressure is created in the xylem that helps to suck water up from the bottom part of the xylem, and the structure itself allows it to withstand low pressures. And I put this in afterwards. I should have put it in the main body, but it's very firm. It has lignified cell walls that are very strong. It's like between a protein, a carbohydrate, lignin, but it's just strong, and it helps to keep the xylem from collapsing. So it's very thin, the xylem, but it doesn't collapse because of that lignified cell wall. And let's see, water travels from the roots up to the leaves in the xylem vessels. Actual transport of ions in the root cells allows water to be absorbed in the roots through osmosis. Yeah, I should have put that. Where is it? The osmosis. Remember, you want to try to get your response within the boxes here. So you don't want to be writing this stuff out here. Try to get it inside that line. Deep root systems allow plants to have a large surface area where water can be absorbed. And remember, we talked about near the surface too, for those quick rains. Thick waxy cuticle. These are kind of the more, structural features that we talked about. Thick waxy cuticle helps to keep water from being released, makes it more watertight. And some desert plants, or most of them, have much smaller leaves, because the leaves are one thing that, you lose a lot of water through the leaves. And they can roll up, let's see, hairs on the surface, and that not only helps to reduce, the speed of air flowing across the surface, which will lower evaporation, but also can help deflect light and reflect it back out. And then some plants use this cam physiology, which very clever, very amazing in evolutionary terms. Instead of opening the stomata during the day when it's hot, there's probably more wind and you got a lot of evaporation, keeps everything sealed up during the day, and then the evening opens them up, and then it can do its final little piece of Calvin cycle and not lose as much water because it's a lot cooler and not as windy during the nighttime. So that is video 2 of 2016 paper 2.